Hi friends, welcome back. Welcome back in JQ Biology. And today I'll be described about the DNA replication of initiation in eukaryotes. Before the first video series of these lectures, I had described about the summary of the DNA replication. That's how a replication takes place in the eukaryotes and which enzymes is involved in formation of the pre-replication complexes in the eukaryotes. So you know that's the initiation steps is the most important steps in both prokaryotes as well as eukaryotes, and also these steps occur in transcription, translation as well as replications. Okay, so the translation, transcription and replication is conserved process in the eukaryotes and prokaryotes, in which initiation is the most important. That's without initiation the cells cannot enter into the cell cycle for the further process or for the further DNA replications, also in case of translation and transcriptions. So details about the initiations in eukaryotes you will be find here. Before going to the initiation steps, you know that the multiple origin of replication is found in the eukaryotes, but a single origin of replication is found in the prokaryotes. Okay, and the complexity of the genomes is much more in eukaryotes in comparison to the prokaryotes. So for simple diagrammatic, for simple understandings, I will diagrams only one part of the segments of the DNA of eukaryotes that's contain only one origin of replication, but you know that the multiple origin of replication is found in the eukaryotes. So this is the origin of replication, that is the specific sequences, specific sites where another proteins binds and help to formation of the DNA bubbles or melting the DNA. This is the ORIs. Okay, where ORC, this is the origin of replication complex proteins, that is the multiple subunits protein from 1 to 6 and have different subunits or domain that's help to another melting and another bind to the specific sequences and another hydrolyze the ATP and get energized. That is from ORC1, ORC2 and so on. So our scientists have detailed studied eukaryotic DNA replication in Saccharomyces cerevisiae, that is the yeast and single cells eukaryotes. Details studied about the Saccharomyces pombe, Saccharomyces cerevisiae and also Xenopus laevis. Okay, so this also ORC differ in case of in case of Saccharomyces pombe, Saccharomyces cerevisiae or Xenopus laevis. Okay, so you will details again that this is the origin of replication complexes, and these origin of replication complexes is not alone responsible for the melting of the DNA. Okay, this is the only required to the attachment of the origin of replication and also hydrolyzing, but alone it cannot hydrolyze. So it requires different sequential attachment of the proteins, that is the CDC6 followed by CDT1 and then finally MCM2. MCM2, that is the many chromosome maintenance proteins, that is the heterohexamers, that is from 2 to 7. This is heterohexamers proteins, that is the load on the strands, but alone it cannot load. First ORC is bound on the early G1 phase of the cell cycles. Okay, and then followed by the CDT1, that is the ORC complexes were followed by the CDT1, okay, CDC6, sorry, CDC6, and after attaching CDC6, you can say that they create sites for attachments of another proteins, that is the CDT1. 2 CDT1 comes on both sides of the DNA. Okay, this is the CDT1 and then ORC, CDC6, CDT1's load the many chromosomes maintenance proteins on the strands. Okay, then what will happen? This is heterohexamers proteins. Okay, heterohexamers protein MCM 2 to 7. These whole complexes is called pre-replication complexes. That the formation of the pre-replication complexes is occur in the late G1 phase. Okay, point out these pre-replication complexes forms in the late G1 phase. That is the initiation stage also involve 3 stages, complete 3 stages. That is the early G1 phase, late G1 phase, and the transition phase. That's after late G1 phase, the timing of entering into the S phase is called transition states. Okay, so alone the pre-replication complex is also in this stance not active. It cannot hydrolyze or melt the DNA sequences, so they require energy. All these complexes require energy. So how energy and where energy come from? That is the another proteins comes, and that is the CDK, DDK. This is the cyclin dependent kinase and DBF4 dependent kinase. That is DBF4 dependent kinases. Both these enzymes come and phosphorylate all the protein. Okay, all the pre-replication complexes. When all protein replication complexes phosphorylates, what will be happens? At this time, the another proteins, that is the CDC6, CDT1, and origin of replication complexes is removed. Okay, these all is removed, and MCM2 that's get phosphorylates, also it gets phosphorylates, and all of these get phosphorylates. Okay, all of these get phosphorylates, and 3 proteins is removed in the late G1 phase at the transition states, and the CDK, DDK phosphorylates all these proteins. After then, it's also not able to melt the DNA sequences. That is the not responsible for the bubbling formations. So another proteins come after phosphorylation in the transition phase and enter into the synthesis phase, and the elongation process is the initiate. That is CDC45 and genes. These both proteins serve for the formation of the replication bubbles. You will see that there is the structures of the replication bubbles, where that is the mini chromosomes maintenance proteins becomes both sides, and that is the genes CDC45 genes. These 2 proteins help to melt the DNA sequences. After melting, create sites for attachment of the polymerases, and you know that the eukaryotic DNA require different DNA polymerases for synthesizing, and prokaryotic DNA sequences only requires polymerase thirds, DNA polymerase third. But in this case, DNA polymerase alpha, DNA polymerase epsilon, and DNA polymerase delta is the mainly replication enzymes that's involved. Here you will see that on the leading strands, only one strands the attachment of the primers requires, but in case of lagging strands, multiple primers is required. Okay, so after attaching the primers on the leading strands, DNA polymerase alpha, that is the DNA pol alpha synthesize, that is the initiator of the adding nucleotide in the both strands, that is lagging strands as well as the leading strands. Okay, so their activity, the limits, their activity has limitations of the adding nucleotides. That's it nucleotides till 30. Okay, it's adding only nucleotides 30 and then hand over to the silent and also DNA polymerase delta. DNA polymerase delta work on the lagging strands for adding nucleotides and DNA polymerase is silent. DNA pol if silence work on the leading strands. Okay, works on the leading strands. And DNA polymerase delta works on the lagging strands. Okay, and these DNA polymerase DNA polymerase delta and DNA polymerase silence takes place after synthesis, after adding of the DNA polymerase alpha, because DNA polymerase alpha synthesize 30 nucleotides and then hand over to the delta and the lagging strand, and on the leading strand DNA polymerase synthesize 30 nucleotides and add the hand over the silence, DNA polymerase silence, and DNA polymerase silence works for the replication or adding nucleotides remaining portion of the DNA sequences. So this was the entering of the elongation steps. I hope that you all have understand about the initiation of DNA replication in eukaryotes, and the next lecture would be the elongation of DNA replication in eukaryotes. So keep watching, and I hope that you like these lectures, and if you like, then please click on subscribe buttons and share for your friends. Okay, so keep watching. Thank you.